Hi folks, it's Jeff here. This is a very interesting Jeff here. I have been working at trying to record this lesson since about 11 o'clock this morning. Started with camera problems. You don't want to hear all this, but I think this is about the 4,000th time I've started this lesson. I'm optimistic it's going to work this time. I've actually gotten through the lesson from beginning to end and had nothing to show for it. So I am a lifelong learner. I'm not the slightest bit upset. I'm highly amused, a little bit frustrated, but that's not the same as being upset. I think part of being an artist is knowing that when you're frustrated, you grow from it. I'm frustrated all the time. I remember when I was 18, if I'd be frustrated with an art project, it'd go in the trash and I'd scream and yell and have a fuss. Now, when I have a problem like that, I think to myself, what a great learning experience. And I grow from it. So I'm not the slightest bit upset. I'm amused, slightly frustrated, horribly. I hate the way I'm looking on camera right now, but there's not much we can do about that. I never was terribly pretty, but I had a nice beard to cover that up. Anyway, for the third time, I'm going to go through this lesson from beginning to end. 5,353rd time I've started it. I think I figured things out. You'll know because you'll see it, which I've said in about 10 of these videos. So anyway, here we are today. Let me get you. Things are not as handy as they were when I started and got everything handy. I'm going to switch to my document camera, which I guess today is my specimen camera. You get to see my hairy arms, or lucky you. I'll show you my tattoo. There we go. So here is a tulip. This is a very stylized, very basic tulip that probably has very little to do with tulips in the real world. But we're going to talk about the parts of a tulip. I think it's really important. I'm not done with my big speech. I'm going to switch back. I know how to do that now. I've learned that today. I want to switch back, and I want to thank Barbara Burkhart, my 7th grade science teacher, for what I'm about to talk about. This is a lesson she presented to me in the early 70s, mid-70s, and it stuck. Barbara Burkhart's 7th grade biology class really provided the framework for science for me for my entire career of teaching. And I've emailed her and thanked her. I couldn't do it enough. Her 7th grade class was about taxonomy, which is how things are named. And the way plants and animals, organisms are named has to do with how they relate to one another evolutionarily, or in terms of systems and their place in the environment. And all those things were discussed, a lot of critical thinking. The only part of that class that doesn't seem relevant today, yet it is, was we had to learn the metric system inside and out, because by law, it was going to be the law of the land that we use the metric system in just a year or two. And we know how that went. This is 40, 45 years ago, and I don't think we're using the metric system. I use it a lot. I work back. We're not going to go there now. So anyway, I want to thank Barbara Burkhart. And I also want to talk to you about why nature journaling is about understanding taxonomy and systems. You need to recognize what you see in the field. You can go out in the field and you can draw that pretty bird you see, or that pretty flower you see, or those shells, or those leaves, or that geological feature. But until you understand how that relates to other things, I think your learning will be rather static. And you can prove me wrong. I'm not going to argue with you. There are lots of ways to learn. And I'm an advocate of every one of them. But for me, I need to understand how things interrelate. And to understand that, I need to know taxonomy, how things are named, and how their systems work and their place in the environment. So we're going to talk about flowers today so that you can do that. Because what I show you today in a daffodil and a tulip is going to be applicable to a maple or a ginkgo or any other flower you see, Dutchman's breeches or a Canadian thistle. They all have these parts in one variation or another. And when you make inferences from what you know, and you put that in your journal, you diagram this picture, draw a flower, and you think about what I've talked about today or what you already know, which is probably much more than I'm going to talk about. You'll be able to make inferences and guesses. Same thing. Inferences are guesses based on the knowledge that you have. And you'll make those inferences, and then you can go back to the studio or get online and you can verify those inferences, or you can find out that you were wrong, which are both the same lesson. Being wrong is probably more important than being right. Being right, you're validated. You're like, oh, that feels good. Hooray, I'm so smart. But when you're wrong, it causes you to dig deeper, causes you to look further into the investigation and to learn more about what it is that you are exploring, in this case, sexual reproduction in plants. This is what made this lesson really fun to me when I was teaching, this was the first lesson where we talked about sexual reproduction. And I never held back ever in my life on talking about sexual reproduction. We didn't get explicit, but sexual reproduction is kind of important to science, especially in 5th and 6th grade. And this was usually the first time students ever heard those words together in a class. And you can use that to motivate. It was fun. Today we're going to talk about sexual reproduction. They get all red and pink in the face, and I'd bring out a plant, and they'd be like, "Oh, okay." But then I'd use explicit terms, and they get red in the face again. Okay, this isn't about me. I'm going to shut up, and we're going to go to the plants. So I'm holding in my hand a tulip, which you're going to see in just a moment. A big yellow tulip. This is a great red and gold tulip I picked up today at the grocery store. And it's blown out because I deprived it of water for an hour or two. I wish I hadn't, but that's okay. It's still a beautiful plant. And let me get this out of the way. We'll come back to that. And we're going to talk about the parts of this flower and how sexual reproduction works in a flower. Then we're going to apply that to 2 or 3 other flowers. And hopefully when you go out in the field, you look at a maple flower, which is nothing like these, and go, aha. Or you can look at the flower of a Dutchman britches and go, aha. Hopefully we can provide some aha moments. Think of a mayapple flower hanging down under those mayapple. You'll see all this there. So anyway, this is a tulip. So we're going to go, first of all, we're going to look at the back side. And this is the stem. We all know that. And most plants have sepals, which are green leaves that are on the back side that cover up the petal while it's forming. We don't have sepals in a tulip. Someone that knows more than I do could argue maybe that these are modified sepals. I'm not sure. But we're not seeing sepals. I will show you plants today where sepals are clear as can be. And then we come up to the top. Let's talk a little bit about symmetry, because it certainly has a lot to do with how you draw. I'm going to stop and check and make sure that I'm recording. I can't freaking tell. Can't tell at all. I shouldn't use the word freaking. I really can't tell. Pause recording, stop recording. We are recording. Thank you. All right. I won't do that anymore. I'm learning the software, folks. Who had to, wants to. All right. So anyway, so we have the stem. We don't have sepals unless these are modified sepals. I wouldn't call them that. And these are petals. And we all know the petals protect the sexual organs of a flower while they are forming. And they also are used to attract pollinators like hummingbirds, moths, bees, the wind. I don't think flowers attract the wind. But many flowers need to be pollinated, physically pollinated, in order for them to reproduce. And we're going to talk a little more about that. Sometimes they're pollinated by human beings, sometimes by the wind. I'll shut up. I'm going on. So anyway, the sexual reproduction organs are down here. I'm going to go ahead and peel these off. I hate to do this. She loves me. She loves me not. She loves me. We're not going to go any further than that. Oops, I just broke the stem. This is going to get very awkward and hard to handle. I've done at least 5 of these today. I'm glad I bought a lot of them. I only bought 1 daffodil, though. We're not going to see that. Okay, so we're getting down. All those outer petals are gone. Now it's time to put my glasses on and really do this right. I'm going to put some of these down here because hopefully you can see. There is loose pollen gathered on this, and I can move it around, but I don't think you'll probably see it on this camera, but just know the little dots of that really dark red black pollen. As an artist, I want to tell you that black never occurs in nature, except in the absence of light. If there's any light whatsoever, there are red blacks and green blacks and blue blacks, and I tend to use a lot of blue and green blacks, but there are lots. Blacks are just really intense color. Anyway, so I'm gonna, I have to pull up a diagram that I want to use because I want to make sure I'm giving some really good standardized language. So those are the petals that we pulled off. Around the outside of this, let me put this on it. I'm used to having a stem and I can't hold this. I'll do that. No, that didn't work either. Hoping we could put it on and all and get a good grip. So around the outside of this, we have the male sexual reproduction organs, and these are stamen. There's my diagram, my cheat sheet. We're going to fill in one later. I just want to make sure I don't make that I give you really, really standard language. So this is the anther and the filament. The anther and the filament make up the stamen. And the anther produces a pollen. If you could see this firsthand, and you're not, I don't think so anyway, but we're going to try. Well, maybe you can. You can see a dusting of pollen. Well, we moved through it. I wish I had a physical way for me to focus that, because that did not work. But there's a dusting of pollen on these, and you can see some sticking to the side of the pistil, the female parts. So anyway, the lower part's called the filament. The upper part of this is called the anther. The entire thing's called the stamen. The anther produces the pollen, and pollen contains the sperm cell. It's not the sperm cell, but it contains the sperm cell. In the middle of this, we have the pistil. The pistil is the female part of the flower, and through pollination, again through physical means with birds and bugs or wind, this pollen is moved from the anther to the pistil. The top of the pistil is sticky so that it receives that. And when the pollen is deposited on the top of the anther, on top of the pistil, the outer coating, that crust, dissolves away, and there's a sperm cell, and actually that sperm cell burrows down through the pistil down to the ovules, or down to the ovary, or the ovules are in the ovary. So we can, I want to make sure again, we're looking about language. So the top of the pistil is called the stigma, moves down through the style, and then down here is the ovary. And it's not very developed when we look at a tulip. So I split one open on a tulip earlier on the 3000th take of this lesson, and there really wasn't much to see. But the sex organs are really clear, that we've got all these stamen around the outside and a pistil in the middle, and that's what you're going to be looking for when you look at plants in the field. Now, that being said, there are some plants, they're all over the board. Every combination you can think of occurs. So there are plants that only have male flowers on one plant and females on the other. Holly is like that, or persimmon is like that, or ginkgo is like that, where you've got a male tree that only has male flowers and a female tree that only has female flowers, and they have to be in proximity to each other in order for them to pollinate. There are other plants like apples where you want to have several trees together because they don't self-pollinate well, but they cross-pollinate. So one apple tree will pollinate another apple tree very well, but they don't pollinate themselves very well. And it takes things like bees. That's why bees become so important in pollination. There are plants that have male and female flowers. Squash and pumpkins have a male flower and a female flower. And in order to get a really good yield of pumpkins or squash, you've got to take a paintbrush and pick up pollen off the male flowers and move it to the female flowers. So there are lots of different permeations of how this works, and that's why you need to have this firmly planned in your head, so when you go out in the field and look at a flowering plant, you can see how it reproduces. I'm thinking of things too, like maple flowers, tree flowers. Some of them are very, very subtle, small. Our maples bloomed 3 weeks ago. tiny, tiny red-black. They're about that color, flowers. The sexual organs are very apparent, but they're teeny, teeny, tiny. And if you're not out there for the 3, 4, 5 days they're blooming, you're not going to see it. So we have the pollen that goes from here to here. The sperm burrows down. Down here in the ovary, there are ovules. Ovules are seeds that have not been fertilized. When the pollen gets to it, they're fertilized and they develop into full-blown seeds. And then many, many trees and plants have different ways that they protect those seeds. We can think about vegetables like peas and beans that grow a husk around them, apples and pears that grow a fruit around them. Some maple trees have the little feathery husks on them. Walnut trees grow a shell around that seed, around those seeds. So you've got lots of ways that the seeds are protected and their longevity is, and that's a whole other discussion, but there are ways those seeds are dispersed, where those seeds are moved that have to do with how they develop or how they're protected or how they're fed. I mean, apple trees and pear trees have a little food packet that goes with those seeds. So I'm going to step away for a moment. Let's see if I can pause this, or am I just tempting fate? I can. Let's see. Pause. All right. So if anything, I'm going to learn more about how to use the software. Here comes Moxie. So I wanted to pull up this one because this one was really nicely done. This is from a lesson before, probably around lesson 20. Just kidding. But I bet we're up to 10 now. So you can see the anthers. You can see the pistil. You can see there's some modified petals in a tulip that actually the anther is part of it. And then down here is the ovary. So this is a daffodil that I pulled apart. And the anthers, the stamen were in a circle around the pistil, and I sliced around the base and pulled that up over the top, leaving just the pistil, which was very, very long. You can see here's the pistil that was on that daffodil. And if you turn over, you can see the ovules. See all those little teeny tiny. Wait, see a little focus. Gosh, I hope so. Wonder if I like to use a lens in the field, and that might help a little bit. You can see those ovules. Those are little immature seeds. Those are seeds that will never be seeds until the pollen, the sperm from the pollen reaches those, and then they will develop into seeds. And I don't know what the fruit of a daffodil looks like or the fruit of a tulip, because we don't think about reproducing tulips and daffodils through seed, but breeders do. That's how you hybridize these plants. That's how you get these really phenomenal daffodils, these phenomenal daisies. I mean, this was beautiful, wasn't it? The red and the orange. Somebody hybridized that. We do things like bulb divisions to learn a lot of asexual reproduction in plants. So bulb divisions and cuttings are the primary way that we reproduce a lot of plants. So that we're involved with it. We don't reproduce them. That we're involved with the reproduction. All right, I'm going to pull down. Let's see what else I have. Let's see. These are my grandmother's bleeding hearts. We moved these she moved these when she moved from Philadelphia to Lancaster, and then we got a cutting from them. And so these are white bleeding hearts. Often you see the pink ones, and they're related to some Indiana wildflowers. And I'm going to just pull back these. You can see the petals are highly specialized. And, these things all evolved for a reason. And as I pull this petal back, I don't know if you can see it, but there's a stamen attached. Yes, you can. I can look at it. You can see there's a stamen is attached to that petal, and to that petal. And if you get right down to the middle, let's see if I can do this. Last time I did it, I got this heart-shaped thing. There it is. I'm going to pull those apart. There we go. And there's the pistil right there in the middle. And so the ovary is down in here somewhere. So I don't want you tearing apart wildflowers. We're not pulling apart wildflowers. I want you to do this with the flowers in your yard, and so that when you go look at wildflowers with a hand lens, you can get down on your hands and knees or take some really good pictures. We're going to talk a lot about pictures in the next lesson on Thursday. Take some pictures that you can go back and use for reference to go do some research. So there we go. There are parts. I've got some pollen gathering there on my drawing board. The last one I'm going to get some violas. I bought all these plants at grocery stores. These are plants that were spent. So they're super cheap. So pansies and violas and Johnny jump ups fascinate me. I think it's a beautiful plant here in Indiana. We have these out. I'm sure all over the country, world, they have these out in the spring while we still have frost danger. So I'm going to make the inference here that I've done a little bit of research about how bees find where they pollinate, and I know there are lines that are invisible to the human eye, and some are visible that lead the bees to where the nectar and the pollen is. And I'm going to guess these might be related to that. And if I were journaling this right now, I'd write that down, and I would go get online and see how do bees pollinate, I don't know, pansies. I don't know. I'd go search some things like that. How are pollinators led to the whatever? I really like to draw pansies because they say they have little faces. I don't really see the face, but it's such a nice geometry. You've got a petal here and a petal here. These mirror each other. This is not so much radial symmetry, though there's some. Bilateral symmetry certainly comes in here. Did we talk? We didn't talk about symmetry. I did the last 27 lessons. So most flowers have radial symmetry. The symmetry comes out from the center like a star. And things like dogs and cats and lizards and human beings have bilateral symmetry. You divide, the symmetry line is down the middle. If I take my body and divide it in half, one side is just like the other, with some exceptions, with some, these are general rules. So flowers are the same, or have radial symmetry. And you could argue this does, because each of these petals comes out of the center. But you also could argue there's bilateral symmetry, because you come up through here and one side mirrors the other. So you have these three petals, and then there are two overlapping petals in the back. These are really fun to draw because you've got these overlaps. You can draw little hat shadows or colored shadows underneath. You got these beautiful lines. Even on this all blue viola, you've got this beautiful white in here and just a little bit of red and that dark blue leading in there. So this is a tiny flower. This is where it gets a little bit hard. But if we pull back on these two outside, and we pull down on the bottom one, actually, let's go ahead and take them all off. First of all, this has true sepals that look like leaves. But you get in here, you can see there's the pistil right there. And you can see itty bitty, teeny tiny stamen around it. I don't know if you can see it here, but you'll see it in the field. Go out, this is a hanging plant. I bought it at the store and brought home, and I can see it. I got lots of flowers. There you can see that top bulb is the top of the pistil, and then that white cluster beneath it up to the northeast to the upper right of that are the stamen clustered around that. So there they are. These body parts, these sexual reproduction parts, these plants all have both parts. But again, some have just male parts, some have just female parts. You'll learn to see those. So let's get out the journal, and we're going to try this one more time. Not the first time I've drawn this today. And I'm hopefully. So I've drawn this really, really basic flower diagram. And again, it's super basic. So, and I'm pulling up this just so I make sure the nomenclature, the vocabulary is what you expect, there we go. So I want to, first of all, this is the stem. I think we all know that. Of course we know that. You buy flowers by the stem. These are the sepals. Sepals, again, protect the bud, protect the young flower, and then they peel back. We had troubles finding them on daffodils and on tulips and on the bleeding heart, but they were very, very apparent in the violas. On this side, I'm going to talk about the female sexual reproduction organs. And in your notes, you can use the female sign. There's no reason to write it out. And that's common practice in science. So we're talking about this structure in the middle here. So the entire structure is called the pistil. I write sideways, so you're going to have to deal with slanting type that's as close to horizontal as I'm going to get it in the way I'm sitting right now. I'm really left-handed. I actually hold papers absolutely sideways when I write. Makes very interesting sign writing things. So the top of this is called the stigma. The middle of it's called the style, S-T-Y-L-E, stigma, S-T-I-G-M-A, style, S-T-Y-L-E. And then this is the ovary. You know, that's a term we all know, right? We can relate to that because mammals have ovaries. And then in here we have the ovules. And the ovules are what become seeds after they're fertilized. And not until. They sit there and they develop and wait till they be fertilized. Then on the male side, we've got the entire structure is called a stamen, the entire piece. Yeah, I want my body parts called a piece. The part that produces the pollen is called the anther. And then this is called the filament. So the pollen is produced here. Some plants, they can self-fertilize, so the pollen goes from here to here. Some plants have to fertilize another plant, so the pollen from here has to go to some tree next door. Sometimes the flowers only have one part or the other. So when the pollen gets to here, the outer coating dissolves, it actually burrows down into the ovules, and then once it does the genetic thing, it shares some DNA, it reproduces in the DNA, and then begins to develop into a seed. And then different trees, as we talked about, develop different ways to protect or feed that fruit or feed that seed, be it fruit or a husk or a nut or a vegetable. Vegetables are actually roots, so that doesn't count. But they'll develop something around that to protect it and or to feed it in the future. I didn't label the petals, but I think we all know petals. So anyway, this is the lesson I've been trying to teach since 11 o'clock this morning. It's now 4:45. This is my 6-hour lesson, which it's been really fun. I've got the tech. I think if you see this, I've got the technology under control, and we can do this much more quickly and much more often. And that was the point today. Today was kind of set aside to do this. So if you have questions, I want to do this live so you could ask questions. And I had a recording in Zoom, so maybe you could have. Maybe next time I'll share that. But until it worked, I wasn't going to do that. If you have questions, email me at gbdavisfolkart@gmail.org. Or if you're in the group, which is Nature Journaling dash the workshops of GB Davis, if you're in that group, you can message me, or you can ask in the group. Wouldn't that be great? Ask in the group under the link for this video, so we all get to see the answers, we all get to talk about it. I encourage you to ask questions that way, because the dialogue in that group is the power of this group. It's not me up here yammering, it's that. So this is what I have to show you. I'll see you Thursday night. There's some things that have come up that I want to talk about. I want to talk about birds, but several of us are taking John Muir Law's bird class that day. So my entire concept of how to teach bird drawing may change. I don't think so, though, since I've read his book and I've been drawing from it for years. But I want to talk about birds for the same reason we talk about flowers today. You get the structures in your mind, you can draw faster, you can see faster, and then you have you can ask informed questions. So I want to talk about bird drawing. I want to talk about cameras. There are other things I had planned beyond this. These are things that have come up. There was one other topic. I don't remember. I will remember what it is. I will look at what we've shared on the Facebook group, and I'm sure other things will come up so that we can talk about ways to make your journal richer. Also, remember to post your drawings. If you post a drawing, your name goes in a hat. I'm going to pull your name out. You're going to get a special gift sent to you. I can't wait to do that. That's going to be great fun. I'm done. I'm going to go home and have dinner with my wife. I want to thank you all for tuning in. Please talk. I'll be on the computer all evening, I'm sure. So if you want to talk to me about this later, that'd be great. Have a great evening. Thank you so much. Bye.